Howdy, this is Lab Report 6, Biochemical Tests, the decarboxylate tests, lysine decarboxylate test, and ornithine decarboxylate test. Group 2, Ana Martinez and
Anna Garcia, Microbiology Fall 2024.
Introduction and Significance. The purpose of this lab is to measure the enzymatic ability of an organism to decarboxylate an amino acid to form an amine with resulting alkalinity by observing color changes. Materials and methods. Lysine decarboxylate test. Separately inoculate the bacteria listed aseptically into the labeled lysine iron agar tubes using an inoculating needle to stab the bud and streak the slant. Incubate and observe for any growth and color changes. And these are the materials. The materials and methods for the ornithine decarboxylate test. These are the materials. Separately inoculate the bacteria listed aseptically into the labeled ornithine broth tubes using an inoculating loop and dipping it into the broth. Incubate and observe for any growth and color changes. The background information, lysine decarboxylase test. A purple slant means LDA negative and a purple bud means LDC positive. Acidic waste products are produced from a small amount of glucose fermentation which lowers the pH and turns the tube yellow. Then the acid pH activates the LDC, causing the removal of the carboxyl group from lysine, which forms cadaverine, and the pH increases, turning the bud purple. Bacteria on the slant will use the peptones to produce energy, thus producing NH3, increasing the pH of the slant and turning the color back to purple. Purple slant, LDA positive. Yellow bud means LDC negative. The acidic waste products are produced from a small amount of glucose fermentation, which lowers the pH and turns the tube yellow. LDC negative bacteria does not produce LDC, therefore the pH will remain low and the bud remains yellow. Bacteria on the slant will use the peptones to produce energy. Um, this produces NH3, increasing the pH of the slant and turning the color back to purple. Port wine maroon slant means LDA positive and yellow bud means LDC negative. Proteus and Providentia (spp) are capable of producing the enzyme lysine deaminase. The product of this deamination combines with the pH indicator in the lysine iron agar and produce a distinct port wine red in the slant, aerobiosis. But the materials and methods, this is the background information for the ornithine decarboxylate test. The turbid purple to a faded yellow purple color means putrescine is produced. When saccharolytic facultative anaerobic bacteria carry out glucose fermentation and acidic waste products are produced, it makes the pH lower, turning the broth yellow. Under anaerobic conditions, adding a layer of mineral oil on the top of the broth, the acid pH will activate the ODC, causing the removal of the carboxyl group from the ornithine, forming putrescine, and the pH increases, turning the color back to purple. Bright, clear yellow color means the glucose was fermented, but no degrade degradation of the substrate ornithine. When saccharolytic facultative anaerobic bacteria carry out the glucose fermentation and the acidic waste products are formed, it makes the pH lower, turning the broth yellow. ODC negative bacteria will not produce ODC, therefore the pH will remain low and the broth will remain yellow. The results and observations. The following slides are the results and our observations of the incubated bacteria in the test tube. The pictures are our results, and the observations are based on what we recorded on our lab notebooks.
Results and observations. Lysine decarboxylase test. For Escherichia coli, there was a purple slant which was LDA negative and a yellow bud which was LDC negative. Purple slant indicating a high pH, yellow bud indicating a low pH. The bacteria does not produce lysine decarboxylase. For Proteus mirabilis, yellow slant, LDA positive, orange bud, LDC negative, produces enzyme lysine deaminase. For Salmonella typhimurium, a purple slant indicated LDA negative, purple bud, LDC positive, purple slant high pH, and the purple bud high pH, so it produces LDC. For Morganella morgani, purple slant LDA negative, purple bud LDC positive, purple slant high pH, purple bud high pH, so it produces LDC. For unknown 102, purple slant LDA negative in yellow bud LDC negative, purple slant high pH, yellow bud low pH, so the bacteria does not produce LDC. An unknown 202, purple bud LDA negative, purple slant LDC positive, purple slant high pH, purple bud high pH produces LDC. For the ornithine decarboxylase test, Proteus mirabilis had an orange color, so a low pH, and inconclusively ODC negative, so it does not produce the enzyme ornithine decarboxylase. But Citrobacter freundii had a yellow color indicating a low pH, so it was ODC negative and does not produce ODC. And for unknown 202, it had a dark yellow color so it indicated a low pH. So it was ODC negative, which means it does not produce ODC. Conclusion. The lysine decarboxylase and ornithine decarboxylase tests are useful in microbiology to help differentiate bacterial species, especially those within the Enterobacteriaceae family and other gram-negative rods. These biochemical tests are significant for bacterial identification based on their metabolic capabilities. Thank you.